Hello, hi, namaskara. My name is Kirtan and I am a second year medical student studying in a government medical college. Welcome to my channel Physiology at Once. Today I will be talking about the topic biliary secretion. Under this topic I will be talking about the bile salts, the difference between the hepatic and the gallbladder bile, the regulation and secretion of the bile secretion. So let's talk about this topic. The bile is secreted from the liver. Its composition is 98% water plus 2% solids. These 2% solids contain bile salts, bile pigments, cholesterol, inorganic salts, fatty acids, alkaline phosphatase, cations and ions. So these bile salts are actually the sodium and potassium salts of the bile acids. These bile acids are again divided into primary bile acids and secondary bile acids. The primary bile acids are secreted in the liver and the secondary bile acids are secreted in the intestine. This bile acid first conjugates with the taurine or the glycine, which produces taurochloric or glycocholic acid. This taurochloric or glycocholic acid is again reacted with the Na plus salts or K plus salts to form sodium taurocholate or potassium taurocholate. This glycocholic acid reacts with the sodium salts and potassium salts to form sodium glycocholate and potassium glycocholate. Hence, these form the bile salts. So what are this function of the bile salts and why it is important? The first one is the absorption of fat. The bile salts helps in the absorption of fat by the formation of micelle. This micelle formation helps in easy absorption of the fat. Next one is the emulsification of fat. Emulsification is nothing but the process of reducing the surface tension of the fat molecules. Since these bile salts contain many surface tension reducing agents, they cause emulsification of fat. It is also a source of bile acid. Since the bile salts are made up of the bile acid plus the sodium and potassium salts, they can again reuse to create bile acids whenever required in the small intestine. It also helps in the absorption of fat soluble vitamins like vitamin A, D, vitamin E, vitamin K. They also help in the activation of pancreatic enzymes like pancreatic lipase. They also help in the prevention of gallstone formation. These bile salts along with the lecithin solubilizes the cholesterol, which again help in the prevention of the gallstone formation. In the exam you can write any 5 of these functions. Now one of the important questions for 3 marks it can be asked as differences in hepatic and gallbladder bile. This is the important question which can be asked as a 3 marks question. So the hepatic bile and the gallbladder bile can be differentiated into 4 points. The first one is the pH. The hepatic bile will be alkaline, whereas the gallbladder bile will be acidic. The water content of the hepatic bile will be 98%, whereas the gallbladder bile it will be 85%. The solids in the hepatic bile they will be 2 gram percentage, whereas it will be more in the gallbladder bile, which is 10 to 15 gram percentage. The bile salts and pigments will be less in the hepatic bile, whereas they will be more in the gallbladder bile. So these 4 points are important and you can write it in the 3 marks question in the examination. Now let's talk about the mechanism of secretion of the bile. It can again be divided into canalicular flow and the ductular secretion. The bile acids and the sodium K plus ATPase function is seen mainly in the canalicular flow, and the bicarbonate and the chloride ion secretion is seen in the ductular secretion. So let's talk about this mechanism of secretion one by one. First, the bile acids is taken up by the hepatic cells from the sinusoidal bloodstream. These bile acids are then binded to the bile acid binding proteins. So these bile acids are binded to the bile acid binding proteins because it is to maintain the concentration of the bile acids in the hepatic cells. If the more concentration of the bile acids happen in the hepatic cells, there will be degradation of the hepatic cells. So upon binding with the bile acid binding proteins, this bile acid is again conjugated with the glycine and the taurine molecules. After conjugation of the bile acid with the glycine and taurine molecules, the micelle formation takes place in the hepatic cells. This helps in maintaining the concentration of the bile acids again. Next, it goes into the ducts. In the ducts, the water and electrolyte balance is maintained and it is same as that of the plasma. Through the duct, it goes to the gallbladder. In the gallbladder, the bile is mainly stored and there is modification of the bile, that is the acidification of the bile. So that's why the hepatic bile and the gallbladder bile is differentiated. So how is this acidification of bile takes place? This is due to the H plus secretion from the epithelial cells. H plus secretion from the epithelial cells. So how does this H plus is secreted from the epithelial cells? So imagine this as the epithelial cells in the gallbladder. The H plus is secreted by exchanging with the Na plus. So the Na plus is taken by the cell and the H plus is secreted, so therefore making the bile acidic. So what causes this Na plus to exchange with the H plus? So therefore there is an Na plus K plus ATPase. Na plus K plus ATPase. So this Na plus K plus ATPase provides the gradient so that the Na plus is exchanged with the H plus. So this happens in the basolateral membrane. This ATPase is present in the basolateral membrane. So that's why the H plus is secreted in the bile, and there is acidification of the bile. There is also absorption of the water in this gallbladder. So therefore the concentration of the bile is increased, that is the bile is acidified, and also there is decrease in the water content of the bile. So this is same as we saw in this differentiation. The bile is acidic in the gallbladder and the water content is decreased. So now let's talk about the regulation of secretion of the bile. It is mainly influenced by the choleretics and the cholegogues. The choleretics are the substances which enhance the secretion of the bile. So directly the secretion of bile is increased by the choleretics. Example is the bile salts, the secretin and the vagal stimulation. The next one is the cholegogues. These are the substances that contract the gallbladder to increase the output of the bile. So there is contraction of the gallbladder to increase the output. Here there is directly the increase in the secretion of the bile, whereas here the gallbladder is contracted more so that the bile which is present already is more increased in the output. So examples of these cholegogues are the cholecystokinin and the fatty acids. The cholecystokinin is the main cholegogues which contract the gallbladder. So that's it for today guys. I hope you understood this video. If you like this video, please like, share and comment, and also don't forget to subscribe to support my mission of making physiology easy, simple and understandable. Thank you.